number 5. So this portion is interesting one. This one I highlighted. The validation of the computationally predicted structure is mandatory. अब ये आगे आंसर भी है क्यों mandatory है. That compares different parameters of the protein three-dimensional structure with that of experimentally available protein structure. जो experimentally available protein structure है, in which we just compare the predicted structure with the available data of the predicted structures with the help of X-ray crystallography and NMR. That an X-ray crystallographic and NMR structure will not be predicted. It is solved structure that has experimental evidences. In computational biology, we usually have hypotheses, opinions, in silico analysis, but in experimental analysis we have evidences. Motivation, model evaluation tools. Okay, first one is ERRAT, very beautiful tool for structure evaluation, and this tool helps to analyze the overall quality factor of the protein. ERRAT uses the algorithm of protein structure verification that especially suited for evaluating the progress of crystallographic model building and refinement. Brief instructions, you know how to use this one, what will be the input file, how to submit your sequence, and then you have the result like this one. Already mentioned that this book was written in 2017 and published in 2018. Now it's 2020. Now the ERRAT result is amended. Now ERRAT showed the results in colors. I remember green, yellow color. Before that it was like black and white. The lines that is in dark black color. These amino acids considered as not the good amino acid overall quality, but the amino acids that are below the line, this line, considered as the good one. We have 82.075% overall quality factor of the predicted structure of RAS-F2. Conclusion, ERRAT evaluation tool was utilized for the evaluation of RAS-F2 structure, and 82.075% of an overall quality factor was observed. Then we have Rampage. It's also a very good tool for the analysis of psi-phi angles and favored region, allowed region, and outliers to find the favored region, allowed region, and the outliers. You guys know how to use this software. And now we are on the results interpretation. Okay, we have result interpretation of ERRAT one. Okay, ERRAT generated a plot indicated the confidence and the overall quality of the model. A single output plot was given by the ERRAT which gives the value of, okay, the results are not much in detail here. You can read any paper that has the results of ERRAT. In discussion section you will find the detailed discussion on the results of ERRAT if you guys are much more interested in the evaluation of ERRAT. Then there are number of residues in favored region. What is favored region? Favored region is considered as the amino acids are at their right position. Suppose there is a serine 29, so serine 29 should be at that position where it is, then it is considered in the favored region. Usually it is considered that favored region should be 98%. And then we have allowed region. Allowed region means that amino acid number 19 is at position A, but it should be at position B. But if it is at the position A, it's okay for that. After that we have number of residues in outlier region, and minimum number of amino acids should be in outlier region. Outlier region is considered as the wrong placement of amino acid. So in this case we have 39 amino acids in outlier out of 326. It should be 1 or 2 or maybe maximum 5. So try to fix the errors. Then we have ANOLEA, in which we usually analyze the z-score and gives non-local normalized energy z-score of the predicted model. This is the brief instruction about ANOLEA, the homepage of ANOLEA. And then we have results interpretation. This type of results we will have for ANOLEA. The summary of the amino acids with the high energy, the total number of amino acids with high energy and its percentage, the total numbers of atoms, the total numbers of non-local atomic interaction, and all that. Conclusion, ANOLEA can be helpful to evaluate the three-dimensional structure prediction. It determines the high energy of amino acids. Then we have mole probability. It's also a very good software. I usually use this one and like this one. It's a bit tricky to use. Now it was user friendly, but now it's a bit tricky to use. Last time I used it, but it has so many good parameters to evaluate the predicted structure. Okay, this is the homepage of mole probability. Then we just submit the structure as we did in ERRAT and Rampage. Instruction of mole probability sequence submission form. Then we have this result. This is interesting result. Protein geometry. Now we have protein geometry. Poor rotamers, we have 54 poor rotamers in this protein. Then we have Ramachandran outliers, that is also 54, 16% almost. Ramachandran favored region, out of 326 amino acids, we have only 210 amino acids. Then we have carbon beta deviation, that should be more than 0.25 Armstrong, and 44 amino acids are here. These are this is not the good result. That's why it is in red color. Otherwise, it will be in green color if the results will be under the parameters of specific tool. Bad backbone bounds 1 out of 2706, and bad backbone angles 100 out of 3654. So result is not seem much good for this structure. Okay, now we have this result for mole probability, and here we also have something in mole probability. Mole probability just nominate which amino acid is in outlier region, like amino acid numbers, amino acid as per gene. This one in outlier region, 0.1% psi angle, and Armstrong is 0.317 Armstrong. Then we have another amino acid threonine, then we have leucine. So it just gives a whole table that describe which amino acid is in outlier region. So we can fix it by Winput software and fix the rotamers. So conclusion is mole probability showed the poor rotamers, outliers, and deviated angles of the predicted structure. So after that evaluation, we was predicted the three-dimensional structure of RAS-F2 protein according to this book, and then we just use different evaluation tools. First, we use homology modeling, threading, and ab initio approaches to predict the three-dimensional structure, and then we have to evaluate the structure. For evaluation, numerous tools are available, and I know you guys use many of them. So we were discussing that for our revision and just to recall our data and just to cover the points that we left. We discussed about ERRAT, we discussed about rampage, then we just jump on mole probability. Today we will study more, and I will try to cover all the 7 chapters in this class, inshallah. So after that, we have Verify 3D, another evaluation tool for three-dimensional predicted structure. Verify 3D also evaluates the predicted structures, and it has the capability to analyze the atomic models with its own amino acid sequence ID by assigning a structured class based on its location and environment. It also compares the results with the library of reliable experimental predicted structures. In actual, the researchers just gather the predicted structure, solved structure by X-ray crystallography and NMR, and they just collect different information and different parameter values and make a database, or you can say a library. And now they just compare the results of your predicted structure with the available library. इसको हम इस तरह समझ सकते हैं आसान अल्फाज में कि उन्होंने एक्सरे क्रिस्टलोग्राफी और NMR से जो स्ट्रक्चर्स प्रिडिक्ट हो चुके हैं उनकी वैल्यूज को एक्सट्रैक्ट किया. सपोज करें उन्होंने देखा सीरीन के साथ प्रोलीन आए तो 0.5 वैल्यू आती है, सपोज कर रहे हैं. आगे प्रोलीन के बाद थ्रियोनिन आए तो 0.7 आ जाती है, थ्रियोनिन के बाद ल्यूसीन आए तो 0.9 आ जाती है, ल्यूसीन के बाद लाइसिन आए तो 0.1 आ जाती है. बैकबोन इस तरह की हो तो ये वैल्यूज हैं, बैकबोन जो है वो अगर टिल्ड है तो क्या वैल्यूज हैं. उन्होंने वो सारी की सारी इंफॉर्मेशन एक्सट्रैक्ट कर ली एक्सरे क्रिस्टलोग्राफिक और NMR स्ट्रक्चर्स से और एक लाइब्रेरी बना ली. अब जब आप अपना प्रेडिक्टेड स्ट्रक्चर उनको देते हैं तो वो कंपेयर करते हैं वो तमाम वैल्यूज को जो लाइब्रेरी अवेलेबल है उसके साथ, एंड देन दे गिव यू ए Z score or other parameters on the basis of these evaluations. I hope you guys got the point. So they just generate a library of the values, and now they are using that values to predict the structure or to evaluate the structure. So this is the home page, and after that, then we have conclusion that the Verify 3D graph shows one-dimensional and three-dimensional comparison of the respective proteins. How the respective proteins, one from the library and one the receptor protein. After that, we have PDB sum generate. It's another tool for three-dimensional structure evaluation. Nowadays, it's may somehow outdated, but you can use and you can study from here if you want to analyze that or use that tool. In this tool, we also have to analyze the Ramachandran plot. Then we have project summary, conclusion of the project summary, in which we have the analysis of different deviation angles of chi angles, and also the amino acid angles. The residue number was given in the original coordinate file. The chain identifier picked up from the original coordinate files that are represented up there, and then you have the values on the basis of you can decide this predicted structure is reliable or not. Ramachandran plot, then we have Ramachandran plot was predicted by different tools last time, and I remember there are so many tools that helps to analyze the Ramachandran plot, in which we have favored region, allowed region, and outliers, and there are so many angles that has been evaluated in the Ramachandran plot. You can have a look here. Three separate columns were given for the three possible conformations of chi1. Then there you have the results of chi2, and okay, this is the all the graphs we generated with the Ramachandran plot. It's in very detail. If you want to study, you can. So now we have another interesting topic here: limitation of structure evaluation tools. That's very important. We have the limitation of ERRAT, like it only calculates the non-bonded atomic interactions having distance more than 2.5 angstrom, and it also has some limitations here. Then we have the limitation of rampage. Two limitations we have for the rampage: one for the verified PD, one for the PDB sum. And here we have so from the limitation section, we have come to know that mole probability has very less limitations, so you can use that. Then we have a guideline for structure evaluation. Okay, here with the same in previous chapters, we have the tool name, availability, description, workflow, URL, and reference. The description section is also the column of description section for all the tools are also very important.